Okay, so there's definitely not Y-linked because, like I said, there's now there's both men and women. So in this case, I would say that it is either X-linked related to recessive gene or related dominant gene. So I don't think it's related dominant gene also, right? Why? Because I said because if you have a dominant gene, then technically everyone will have it already. No, doesn't make sense also, right? So I would say that. Let me see. Yeah. Just want to make sure in case I get it wrong. Okay, so it is actually an autosomal recessive gene. So the answer is C. Okay, it is not X-linked, it is not Y-linked, but it is actually a recessive gene on the autosome. But this can be inherited by people, so which means parents can pass down to their offspring. Then the offspring have a chance of getting it. Okay. Number 5, which of the following processes is an environmental factor which causes variation? All right, they want you to talk about environmental factors. So in genetics, there are two kinds of factors that can affect mutation, or it can actually affect the different traits that's being produced, which then creates what we call as variation. Okay, so there are genetical factors and there are environmental factors. Sometimes both of them can be together, but sometimes they are separated. All right, so let's look at the first one. Which of the following process is an environmental factor? So definitely not the first one. Crossing over is a process where, if you remember, right, crossing over is when you have two of the chromosomes during meiosis, they overlap each other and then they basically exchange a part of the genes. For example, you have two different chromosomes overlap with each other, right? And then this part that they overlap, they will swap. So after that, what happens is that after crossing over, one of the chromosome may reshuffle its genes, and for example, you will get an exchange, I would say an exchange part of the genes. So example, if they overlap, right, then they will basically exchange a part of their chromosomes, and then it becomes a different variation already, different variant. So that's called crossing over. And it only happens during meiosis, which is the separation or the production of reproductive cells. Gene mutation is caused by genes also, defective genes, right? Like for example, like I said, maybe you didn't born with diabetes, but after you grow older, you eat a lot of sugar, you suddenly get diabetes. So that's actually gene mutation because your body stops producing insulin. But in this case, this is not linked to the environmental factor. It is more of a genetical factor. Adaptation, yes. Adaptation means, for example, I think I'll explain to you this phenomena. Okay, I didn't explain to you this phenomena. I'm going to explain to you this phenomena. Hopefully, I'll be able to give you an idea of how this actually works. Okay. So I'll tell you a story, and hopefully you'll be able to understand how it works. During the Industrial Revolution, okay, last time in the I think I'm mistaken, it's the Western country. All right. If you read your Sejarah book, right, you remember there's something called Industrial Revolution, which means, I think you guys call it as Revolusi Industry, Revolusi per Industrian. So what happened was last time, right, when there was a rise of machines, when human beings started to create machineries and things like that, all these factories that they produce and all these manufacturing plants and power plants with burning coal and things like that, it pollutes the environment with smoke. Okay. So what happens was last time they had this moth. Let me show you. White peppered moth. Okay, so last time, there were two types of this moth. I'm not sure if you guys seen this moth before. I think Malaysia don't have much of this stuff. But sometimes you come to your house and you can see this moth flying around. And then people will tell you, like for example, your parents will tell you that, there's a relative who visit you, because there's a lot of this kind of superstitions, and where my parents also tell me that, oh, when a moth comes inside your house means there is a relative that visit you from, last time when you're dead. So these are called white peppered moth and black peppered moth. The reason why they're given this name is because when they fly and when they flap their wings, right, some of the powder from their wings will drop off, and it looks like pepper grains. Okay, so they're called white pepper moth and also black pepper moth. So last time, during before the Industrial Revolution, there was actually more white color moth than black color moth. There are different types of moth, and this species had two different colors. Okay, so we have the black one and the white one, but the white one has we have more of the white one than the black one. Okay, so I explained to you why. Now, last time, right, when they live in the forest area, most of the background are whitish in color. All right, for example, they can camouflage with the background. I would say the tree barks, the leaves, and everything. So white pepper moth are usually targeted by birds. Okay, so what happens is that the white ones will survive better than the black ones because the black ones cannot camouflage. They can try to hide behind like tree barks and flowers and things like that, but birds can see them very easily. So birds eat most of them. So that's why you can see here that at a time before Industrial Revolution, there are more white moths than black moths. Okay, then after that, human being came about, and then they start to, create all these factories. They start to use vehicles. They burn a lot of fuel, and when you burn a lot of fossil fuel, right, there's a lot of smoke coming out. So this smoke basically covers up the forest area with a thick layer of dust. All right. So slowly, right, you can see the plants are covered with dust, flowers are covered with dust, the ground is covered with dust. So the background becomes darker and darker. Okay. Now, when that happens, right, what happened was the black one could now camouflage better than the white ones. So the birds can see the white one easily compared to the black ones. So what happens is that most of the white color moth were then eaten by the birds, and their population becomes lesser and lesser. But the black one can survive better. Because why? The black one can multiply more and more, and it can camouflage the background. So that is what I call as adaptation. You understand? So we have adaptation means, right, if the species can survive in environmental change, right? We can see that ever since the Industrial Revolution, our air is very polluted. So what happened was the more, I would say the stronger species or the stronger kind of living thing which can always survive all this change in environment, they are the ones that will be chosen for natural selection. That's why you can see over here, these two peppered moth are often used in genetics. When they talk about adaptation, right, they're talking about this. All right, whichever species or whichever color variant that can survive better in the environment will multiply and basically survive better than the other one. All right, so that's an example of adaptation to you lah. So you can see like this, for example, right? Okay, last time you can see this white color pepper moth can always camouflage to the background. So what happens is that here you can see there is a background which is like tree bark. They can camouflage their wings because it looks something like the tree bark, and the birds cannot really see them. But the black ones you can see, right, is very easily visible. So the birds will always eat them. But after that, slowly, you can see, right, after the Industrial Revolution, let me see if I can get you another picture, like this, for example, is here. After the Industrial Revolution, when they have black color, thick smoke and dust, the black color can then camouflage better than the white ones. So the white ones are normally seen by the birds, then they'll get eaten very, very easily. Okay, so that's the variant I can show you lah. Yep, very simple examples that I can show you, right? So this is a story that I forgot to tell you last week. I'm gonna explain to you this, but I forgot. But it's a good thing lah. At least you'll learn, right, how it works. See, this is a comparison. The black one and the white one after Industrial Revolution. The black color ones can camouflage better than the white ones. Right, so that's what I meant by adaptation. And again, chromosomal mutations also caused by genetics, so that's definitely not the answer. So again, environmental factor here means adaptation. All right, number 6, the diagram below shows an example of a chromosomal mutation. We have an extra XXY. See that? You have an XXY chromosome. This is actually a guy. This is actually a guy with an extra X chromosome. So as long as you see an XY, right, it tells you that it is actually a male. But if we have another X in front, it means that it has this extra X, which is a chromosomal mutation. Okay? So it is not thalassemia and it's not hemophilia. These are all gene mutation. Only Turner syndrome or Klinefelter syndrome is caused by chromosomal mutation. Now, the answer is Klinefelter. Why not Turner? Turner syndrome is for girls, which we have something called XO. XO means girls are supposed to have 2 X chromosomes, but end up they have only 1. You see that? So when you have 1 X chromosome, you're lacking of the other one, so you have Turner syndrome. Remember that? So for Klinefelter syndrome, it is actually XXY, a male with female characteristics. That's what Klinefelter syndrome is. Okay, all right, next 7, the diagram below shows the stages that occur during the process of mitosis, all right? So we have P, Q, R, and S. So remember, I told you how to see, right? Very easy one, okay? When you see the nuclear membrane disintegrate, that's the start of mitosis. Again, what is mitosis? Mitosis refers to cell division of somatic cells. So somatic cells are basically cells which are every single cell in your body except for your reproductive cells. And reproductive cells is basically meiosis, if you recall before. Right? So how do we know which sequence it is? I think normally they ask you this question in exam. They will ask you to arrange them in sequence, right? Can see here, which of the following is the correct sequence to show the stages in the process of mitosis. So they want you to know the sequence. Actually, very easy to see when. I remember I told you that the first step of mitosis is there must be a nuclear membrane disintegration. So there'll be R. How do I know? You can see there are dotted lines in the nuclear membrane. This one over here is called the nuclear membrane. It is a part of the nucleus. Okay, so R is actually the first step. So I'll put here R first. Next, you also see there is a spindle fiber formation. So this is actually called a spindle fiber. No, sorry, it's not spindle fiber. Spindle fiber is this one. Spindle fiber. And then this is actually the centrioles. Right? So after that, next one you can see is that we will start with P, remember I told you? So we have PMET. I explained to you as PMET here. PMET means prophase to metaphase, prophase to metaphase, to anaphase, and then to telophase. Okay, so R is technically your prophase before your cell starts to divide. Of course, there will be replication of DNA and things like that. So next one we look into is M, right? M is metaphase. How will you know which is metaphase? Very easy. I always tell you metaphase is when you see all of the chromosomes aligned in one straight line, P, right? All of this chromosome is aligned in the middle in one straight line. We know that now this is metaphase. So we have R going to P. After what happens is that there will be anaphase. Anaphase is when the chromosomes are dragged to opposite poles, which is Q. See, right? They are dragged to opposite poles. One part of the chromosome goes to the right and the other part goes to the left. All right? So the next one will be Q. Then after that, you can see over here S. S is when there is a what we call a cytokinesis, where there will be a cleavage formed between the two cells and they'll start to divide into two different sites. All right? So the last one is actually S. So your sequence should be R, P, Q, and then S. Okay? R, P, Q, and then S, B. This one you have to read, you have to read back and see, revise back. If you don't remember, you have to revise back. No choice. All right, number 8, what happens to the number of chromosomes of an individual with Turner syndrome? So we have Turner syndrome, remember? What is Turner syndrome? I'll explain to you just now. Turner syndrome is XO. All right, so we have XO. So it's a female which lacks one X chromosome. Right, the individual has low number of autosomes. Is that true? No, autosomes refers to the chromosome number 1 to 22. Okay, autosomes is a chromosome number 1 to number 22. All right, next, the individual has low number of sex chromosomes. Is it true? Yes. All right, why I say correct? Because you can see, right, XO, XO means that's one lack of X chromosome. The X chromosome is considered as a sex chromosome. So B is the answer lah. Okay. C has one extra autosome. That's wrong as well. Usually, if they have one extra autosome, is usually Down syndrome. Okay, Down syndrome means they have extra plus one more chromosome number 21. Okay, chromosome number 21 is for Down syndrome. And then D, the individual has one extra sex chromosome. That's also, like, for example, XXY. That will be Klinefelter syndrome. Of course, there's one more called XYY. So XYY is the psychopathic tendency. Remember I told you that one, right? So that's XYY. But in this case, we're not using that. So just remember, there are XXY, which is called Klinefelters. And then there's also XYY for super male. We call them. Super male doesn't mean it's Superman. All right. It's basically a male with psychopathic tendencies. Tendency only, not necessarily you become psychopath. Okay, but there is a tendency there. Number 9, which of the following is true about color blindness? So, like I told you before, color blindness, there are two kinds. The most common one is called the red-green color blindness. All right. So let's look at the answers over here. It usually happens to women. Is that true? I wouldn't say it's not. It is not usually happening in women. All right. Just double check and see whether it's correct or not. I mean, double check. Yeah. Color blindness. Because there are two types. Let me see the other types. Okay, all right. So color blindness, right, is actually affected towards the X chromosome. There's a huge relationship between color blindness towards the X chromosome itself. All right. So I would say that technically, it is a recessive mutant gene that are located in the X chromosome. All right. So the answer is donkey instead. So in this case, we'll say that if the individual is a guy, the guy will receive, if the guy receives the X chromosome with the recessive gene, then the guy will be colorblind. For women, they have 2 copies of the X chromosome, so they must have both of the recessive gene, then they will get it. All right. So I would say it is more common in guys compared to girls. That's why you see A here is wrong. All right. It doesn't happen to women usually, but it happens to men usually. Because why? As long as men inherit the X chromosome with the colorblind gene, then automatically the man will be colorblind. Because why? Only got one X chromosome. But for females or for girls, you have 2 copies. And there's a lower chance for you to get, because I would say that you'll have to inherit 2 copies of the recessive gene in your X chromosome in order for you to get the disease. So I think that's why I said that it is more common in guys rather than in girls. Okay. All right, so that was basically the objective question. So I actually added some extra explanation for you so you can understand better. All right, so is there any question you want to ask based on paper 1? No? All good? Okay, cool. All right. So of course, if at any times you want to ask any question, right, you just ask me. Okay, if you don't understand or anything, I can help you out. All right. All right, so let's move on to the next part. Let's look at it. Let's see. So this is actually very familiar, right? I've actually showed you this picture before. We're gonna look into this. There is something called amniosynthesis, which I explained to you before. So amniosynthesis is a condition where we try to extract the fluid inside the baby to see its karyotype normally. Okay, so karyotyping is a technique where we look into the chromosomes that they have and try to see if there's any abnormality in terms of chromosomal mutation. But in this case, there's also and many other uses for it, but the main use for that. So you can see here, the diagram below shows that amniosynthesis method. State two importance of this method. So the first importance, I would say that, like I said, because mainly amniosynthesis is done, right, because we want to know the baby's karyotype or the fetus karyotype, the karyotype of the babies or fetus, basically. My baby, baby is when you born, haven't born means it's fetus, okay. Yeah, so I think better choice of word is fetus, Okay, so if you don't remember what karyotype means, karyotype shows you the complete set of chromosome. Okay, complete set of chromosome. So when you look into the karyotype of a particular living thing, right, or like in this case, this fetus over here, what you're going to notice is that you will be able to see all of the chromosomes that they have inside, right? How do you do is that normally, right, there's a lot of cells inside the amniotic fluid. So when you extract some of the fluid out, inside the syringe here, we have some of the cells as well. So then they can basically take one of the cells and then look under the microscope and see basically what kind of chromosomes that you have. So in this case, I would say one of the importance here is to determine the gender of the fetus, okay? Okay, so last time, how parents determine the gender of the fetus is by waiting till it develop large enough to see its genital area, the private part, right? If you have it's either a male or a female, right? So by looking at the genitals, you'll be able to know, okay, this is a boy or this is a girl. But now not really. Now, right, the moment the fetus is formed, the doctor is able to just take out the fluid from the amniotic sac, and then they can immediately see the chromosome itself, so they will know very easily. Either you are 22 plus XX, 44 plus XX or 44 plus XY, right? So if they determine, like, for example, if you are 44 plus XX, then definitely it's a girl. We have 44 plus XY, that will be a guy. So I would say that one of the most important part of this method is to determine the gender of the fetus, okay? Secondly, we can also use it to determine, any chromosomal mutation. In the fetus, okay? Which could potentially lead to diseases, okay? Like, for example, sometimes parents want to know if their fetus are okay or not by looking at this chromosomal mutation, see whether there's any chromosomal mutation, right? So maybe, for example, if I see the karyotype of this couple's, this couple's fetus karyotype, okay? So I'm going to show you how it looks like. Karyotype Down syndrome, for example, okay? So what you will often see inside your exam paper, right, like an SPM, is that sometimes they may give you this kind of karyotype. It's quite blurred, this one. Let me change to another one. Okay, so you can notice here, right? This is a karyotype of a person with Down syndrome. So in medical terms, sometimes we call Down syndrome as trisomy 21. The word trisomy 21 basically means that he has an extra copy of chromosome number 21. So please do remember this image. This is called a karyotype by looking at the chromosomes and how many that you have for each pair. Usually, for a normal human being, you will always have only a pair of each chromosome. All right, so you can see over here, right, number 21 here, they have 3 copies. So we know that this person has Down syndrome. Okay, so we can actually tell whether this particular individual is this baby that inside the womb, right, is developing, whether it is Down syndrome or not. Okay, so a lot of this, I wouldn't say that the parents will look at the karyotype and then see that the child is Down syndrome, then they'll immediately go for abortion. It's not that reason. It's just that sometimes couples want to know if their child is healthy, whether is there any kind of chromosomal mutation that can lead to diseases. A lot of cases where even though parents know that their child is Down syndrome, the child has Down syndrome, they will still continue, the development process. And then when they give birth to the baby, they still take care of the baby. They still go raise it up. There's nothing wrong technically with being Down syndrome, because if you can see a lot of documentaries in the internet, there are a lot of Down syndrome individuals which can grow up a normal life. Okay, they may be slightly different than us in terms of looks. They may be a little slow when it comes to learning, but they can survive up to old age as well. They can carry out normal activities just like us. It's just that, like I said, they might need a little bit more time to learn the same skill as a normal person. That's it. Okay. So by knowing this karyotype, you will then be able to tell if the person is abnormal or normal. Right. Another example I can give you is basically a karyotype of, say, Turner syndrome. So a Turner syndrome's karyotype would be like this, for example, right? You have 44 chromosomes in total. Yes, done, normal. These are all autosomes. So you see number 1, number 22, right? Number 1 to number 22 are called autosomes, while the rest of the last 2 is called the sex chromosome. So we see over here, this is actually a girl, supposedly this is a girl. No Y chromosome is a girl, but the girl here actually has 1 less X chromosome. So this is called Turner syndrome. All right. So when you have a lack of X chromosome, chances are is that this person will experience stunted growth, and possibly the person will die at a very young age. I would say it's one of the main symptoms of Turner syndrome. But like I said, the purpose of this is to look into the chromosome and see whether you are okay or not in terms of karyotype. Okay. Of course, there are many others lah. So I'll show you some examples of it. So again, the importance of this method is actually to determine the gender of the fetus and also to determine any chromosomal mutation in the fetus. All right, based on the diagram above, name the sample taken. So I would say the sample taken will be the amniotic fluid. Okay, so the answer here is the amniotic fluid. So amniotic fluid is the one inside the amniotic sac, the one that makes the baby float inside. All right, C, state the method used to separate the sample and the fetus cells. Okay, I think it's what it means is cryonic villus sampling. Wait, let me double check the name. Cryonic yeah, I don't know how to spell it. Cryonic villus sampling. Okay. Let me just double check the spelling. Unless I may spell wrongly also. Okay. It's called CVS, correct. So I found the spelling. I also sometimes quite confused with it. So it's called chorionic villus sampling, or in short, it's called CVS. Okay, so it's a bit hard to memorize, so please just try to memorize it. Okay, chorionic villus sampling. Okay, so if you were to look up on the internet, right, let me show you. If you were to type chorionic villus sampling, it will show you this image. Okay, you see, right? They have this needle which is stuck inside, and then they'll pull out, basically they'll sample out the amniotic fluid from inside the amniotic sac itself. Okay, so there are two different types, I would say. This is actually called amniosynthesis, and there's also chorionic villus sampling. Alamak, so hard to pronounce. Okay, so that's how you do it. State the technique used to detect the fetal position and movement. So this basically what it means is that how do you actually view the baby? Okay, so when couples want to know how their fetus is doing or how it looks like, I mean, technically you can't really see how it looks like in real life, but you can roughly see the figure and the shape of the fetus. All right, so it's called ultrasound technique. Okay, ultrasound technique. So what does the ultrasound technique does is that it basically it's a technique where they use ultrasound to view the shape of the fetus. So I'm pretty sure you see this a lot in movies, okay? So right, now you can see this kind of baby. So how it works is like this, they'll normally put some gel on top of your tummy, on the belly area, on the womb, and then they'll use this detector, this machine that produces ultrasound, and it will scan the inside of the womb and see how the baby looks like. So after what happens that you can see over here, you have something like this as an image. Okay, you can roughly see the shape of the fetus. You can't really tell for sure how it looks like exactly. All right, but sometimes you can see the fetus can move inside the tummy and whatnot. Not tummy, sorry, the womb. Okay, so this is how it looks like. So it's called ultrasound technique. All right, next, Mrs. Suraya is in her 18th week of pregnancy. Okay, so some of you may not be able to judge. We know you all understand that I explained to you last time it was 9 months of development inside the mother's uterus, right? Okay, the womb area. So here, what about 18 weeks? So very easy to know, lah. Okay, we know that 1 month, right? 1 month, there will be about approximately 4 weeks in total, right? Okay, so 4 weeks in a month, and therefore approximately like 4 weeks in a month. So you have like 18 weeks means you try to 18 divide by 4 and see what you get. Okay, so 18 divided by 4, you won't be able to get a nice number. It will be around like 4 months plus. Okay, 4 months plus, maybe around 4.5 months, I would say. So this is halfway through your pregnancy. Okay, halfway through the pregnancy of Mrs. Raya. So Mrs. Raya say after she has developed the baby for 18 weeks already, she wants to do amniocentesis. Okay, can Mrs. Suraya undergo this procedure? Can or cannot? So let you all think about it. Can or cannot? Technically, I wouldn't say it's a can or cannot. Actually, can. All right, I wouldn't say cannot also. So can technically. And they did not ask you to explain anything further. So basically just 1 mark. So let's put as can. Okay. Right. Number two, the diagram below shows the genetic engineering technology. So I did explain to you how this actually works, right? This is a way to get insulin in a very cheap way. So I remember explaining to you that last time, if a diabetic patient wants to use insulin, they'll have to get it from animal sources. But in this case, now because we have genetic engineering, we're able to manipulate microorganisms to basically do the job for us. They'll produce a lot of insulin for us in a shorter period of time without any animal cruelty involved. Okay. So in this case, how do we do it? Very easy. Like I said before, we take a healthy person's insulin gene and incorporate it inside a plasmid DNA, right? So again, this is plasmid over here. Plasmid is actually a circular DNA inside bacteria, okay? Not to be confused with its own DNA lah. There's a chromosomal DNA over here, and there's also a plasmid over here. So that have bacteria normally have two different types of DNA. Okay, so we have the insulin gene incorporated into the plasmid. And then why is it called a recombinant DNA? Is because we integrated the insulin gene inside the recombinant DNA. So we are basically mixing the insulin gene into the plasmid itself. Okay, then after that, we will go towards the recombinant DNA. So once we get this plasmid combined together with the insulin gene, right? This is your insulin gene, by the way. And after that, they will incorporate it inside the bacteria, forcing the bacteria to take it in. And then the bacteria itself will multiply into many copies, right? We know the bacteria can grow very fast. So they can multiply, become 2, 2 become 4, 4 become 8, 8 become 16, then 32, and so on. So they grow in a large number, millions and millions of cells overnight. And then they will also replicate the gene. Then after that, you have a lot of copies of the bacteria containing the insulin gene, and then they will be placed inside a fermentation tank where they will basically live happily with all the nutrients they can get. But at the same time, they also produce the insulin that we want, right? After that, we separate the cells from the solution, purify the insulin, and then we put it into a bottle, which can be used to inject it into a diabetic patient to get insulin. So you can see over here, what is the meaning of genetic engineering technology? So I would say in a very simple term, genetic engineering is when we remove the gene from a living thing and place it inside another living thing. So how do we actually phrase that? Let's see. Okay, I would say inserting a gene from an organism into another organism. So that is known as genetic engineering. Okay. So again, initially we saw that the insulin gene over here is from a human being, but after that we place it inside the bacteria. Bacteria's plasmid, right? So that's called genetic engineering. We're basically inserting a gene from a living thing to another living thing. Right now, based on the diagram above, what is R? So R here is known as a recombinant bacteria. Okay. Again, R is called a recombinant bacteria. It is also considered as a genetically modified organism. All right, state the function of the fermentation tank. So fermentation tank is basically a part where the bacteria is able to grow and multiply. Okay, so I would say here to allow the bacteria grow and multiply. So just put a comma here, I think, to grow, multiply, and to produce insulin in a large amount. Okay, all right. Now what is process K? So you can see over here, once they produce all the insulin really, right, before it can be converted to pure insulin, uh, we know that inside this fermentation tank, it will produce a mixture of liquid, right? Basically containing the cells or the bacteria, contains some byproducts, contains some waste material, leftover sugar and things like that, right? So we don't want all of those things. We just want the insulin itself. So I would say process K here is actually purification. Okay, purification process. So purification process is when we purify the insulin and make it like pure, so that we can basically like we can purify it, then we can use it as a whole thing. Okay, next, what type of human cell is taken to obtain insulin gene? So actually, right, I did mention to you this before. The human cell that we take from is actually from the liver itself. Okay, so just to let you know, right, where this insulin is being produced. Insulin is produced by a section of your liver called the islets of Langerhans. Okay, so we have a liver, right? We have the liver, and then we zoom in some more. We can see a section of liver, which is called the islet of Langerhans. And we take the insulin gene from the cells inside the islet of Langerhans. So I'm not sure if I should write liver cells or should I, no, sorry, not liver, pancreas. My bad, sorry. Just change that, yeah? Pancreas. Liver blood. Pancreas. Okay. So I would say, okay, like a two answer lah. I would say that this islet of Langerhans is more for biology students. So maybe you shouldn't remember this. I mean, it's a good thing to know. It is a more or less a general knowledge for you. But in your exam, I think just put pancreas cell. Okay, so cells from the pancreas. Okay, so below the liver, there is a organ called the pancreas. So the pancreas over here produces the insulin. all right, that we need to convert sugar to fats. And specifically, there's actually a section of the pancreas which contains these cells called the islets of Langerhans. So they are the one that produces this insulin gene. All right, I have to take one advantage of insulin production. So state one advantage of insulin production. I'm not sure if they're referring to the insulin production, the process. All right, are they referring to the process, or are they referring to bacteria as a host? Okay, so I would say that if it's the process, it will be. And I give you two. I give you two answers. All right, state one advantage of insulin production. I would say that of course the main purpose is actually to help to um can be used by diabetic patients to produce to convert sugar to fat. Okay, but I think most likely is referring to the process of producing insulin through bacteria lah. So meaning to say, okay lah, we'll put it here as more insulin can be produced in a shorter amount of time to be given to people with diabetes. Okay, yeah. Okay, next, number three. The graph below shows two types of diseases which are caused by mutation. So we have, actually, if you see properly, you already know lah, because I give you this picture in my notes, right? So this is actually called Down syndrome. I know P, you can't really see it, but can you guess what disease P is? Q is definitely Down syndrome, so I'm gonna put here Down syndrome. When you say Down syndrome, it's not Down syndrome, it's Down's syndrome. It's actually a person's name. Can someone tell me if you can guess what P is? This by looking at diagram. Anyone? Just make a wild guess for your one. No? Nobody? On track? Okay. If you see properly, right, this guy's hair is actually white in color, which tells you that it is actually albinism. Huh? I know I can't really see it much, but if based on the hair color, you can see here, it's a very, I would say it's a very good reflection of white color. So I would say that P is actually albinism. Huh? Albinism. Right, state the type of mutation for P and Q. P is actually a gene mutation because it is caused by the gene that is not producing melanin. Okay, so I would say P is a gene mutation, but Q is actually called a chromosomal mutation. Oh, you thought he's old? No, it's not old. I mean, yes, he's older, but if you see the hair properly, it's actually whitish color. Supposedly, if there's a color, then you'll be able to know lah. But there's no color here, so you can't really tell. But it's actually, yeah, it's actually albinism. I'm not sure it's called albinism or albinism, you know. Let me double check and see. Because they're called albinos. Albinism. Oh, it's albinism actually. It's not albinism. Sorry, wrong spelling. Change that. Albinism. Yeah, I thought it was weird because we call them as albinos. Okay, people with albinism or albinism they're called albino. So yeah, I think it's better to call them. The disease is called albinism. Yes. Okay. Next, what causes individual Q to have the characteristics shown in the diagram above? So it's asking you what causes Down's syndrome. So you can put here an extra copy of chromosome number 21. An extra copy of chromosome number 21. Okay, or you can also say 3 copies of chromosome 21. Both also acceptable. Right, the state one factor that causes mutation. So there are many factors that can cause mutation, all right? But I think the easiest one will be exposure to radiation. You can also say exposure to mutagens as well. So I give you a very simple one to remember. The first one is called exposure to radiation. Or exposure to mutagens. So if you don't know what mutagens is, mutagen is actually a substance, a chemical substance which are carcinogenic. So they can actually cause cancer. All right. That's one of the main reasons I give you. How do we explain it? Okay, next. Section C. The diagram below shows 2 types of cell division. we have mitosis and we have meiosis. Okay, so we know that basically mitosis happens in the somatic cells. And then the other one is happening in the reproductive cells. So gametes, like sperm and ovum, basically, right? So in this case here, state one similarity and 5 differences between the 2 types of cell division. So I think very easy ones will do. So let's just put here. Okay, so there are, I did remember telling you to memorize the examples of the similarities and the differences. So I would say that between mitosis, we should take a while. Let's see here. Okay. Mitosis and meiosis. So I'll give you the first one first, similarities. So what's the similarities between the two is that it is a cell division, a type of cell division, okay? A type of, okay, how about this? The easiest one, to produce new cells. Okay, to produce new cells. Okay, now we come to the differences. Okay, so we're gonna go through and look into the differences between the two. So the differences is basically like this. So you can compare side by side, like easier. Okay, so number one, I would say the most obvious thing is that one of the things you remember is the number of cells being produced. Okay, so remember last time I told you mitosis produces 2 daughter cells, but for meiosis, it produces 4 daughter cells. Okay, so the first differences that we can see here is that number one produces 2 daughter cells. And then you can put over here, produces 4 daughter cells. All right. Then, of course, the second one, what else can we think about? Daughter cells are identical. All right. And the second one basically here will be daughter cells are not identical. Sorry, daughter cells are genetically identical. Change that a bit, okay? Are genetically identical. The other ones are daughter cells are not genetically identical. Are not genetically identical. Okay, so the third one is that mitosis, cell division happens only in one cycle. Okay, and for meiosis, cell division happens in 2 cycles. For those of you who are confused, why do I say a 1 cycle and 2 cycles? So if you recall back last time when I told you about the cell division process, right? The Mitosis part, you can see that they undergo PMET only once, which is basically from prophase to metaphase to anaphase to telophase, one time only. They produces two identical daughter cells. But for meiosis, what happens is that there will be two cycles in the cell division. So what are the two cycles? It goes through one time of PMET first, along with, of course, the crossing over part. And the second time, it goes through PMET for the second time, right? So that's why it has four daughter cells at the end. Okay. So one more significant part about this is that the main difference that both of them have is that there will be no crossing over in mitosis. Okay, no crossing over between chromosomes. But in meiosis, crossing over happens between chromosomes. So these are the differences between mitosis and meiosis. On the left side, it's mitosis. On the right side, it's meiosis. So they ask you for, wait, how many was there? 5. So recall back, let me see for a while. Oh, they want 5 differences. Sorry, they want 5 differences between that. So we got one more to go. So we have 4 differences already. You need one more to go. Let's see if I can think of another one, if I can recall back. Oh, yes, of course. Okay. So you can say here, for mitosis happens to all somatic cells, but not in reproductive cells. And then number 5 over here for this one happens to only reproductive cells. So yeah, there you go. We have one similarity on top, which explains to you the differences between mitosis and meiosis. All right. And then we have the differences between mitosis and meiosis, 5 different ones that we know. All right. So if you can, normally in the exam, they won't ask you to compare so many traits. They will probably ask you 3 differences and 1 similarity, which can basically give you 4 marks in total. All right. Now, if you ever come across such an essay question, right, you don't have to write in the paragraph form. All right. Like I explained to you many times before, you don't have to write in paragraph form. You can always do this like me, for example, right? In this case, I draw a table-like, I would say table-like, okay? Don't scribble too casually. I would say try to write in the proper way. This is actually considered as proper. So maybe what you can do is you can draw a table instead to list down the similarities and the differences. All right. I would say it will do your examiner a very good favor because why, like I said before, think about this. The examiners of SPM for science subject or any subject also, right? They have to mark like thousands of paper. All right. Not thousands, probably hundreds of paper. Okay. So hundreds of paper, imagine this, you have to basically mark so many papers. So just to make the life easier, I would say that in essay questions, just make the life easier. Put very clearly, write your points very clearly, avoid too many words. All right, go straight to the point. And I like very much if you can put it into a table form for comparison questions like this. Okay, you can see, right? It's all comparisons. I put it side by side. Of course, I can't really draw a table here because I don't have a ruler to draw, right? But if I could, I would draw a table. And I would also basically put everything nicely lah. Okay, maybe box it up lah. I would say box it up. Okay, and then I'll draw a table at the end. That'll be nice. Okay, just to make a comparison wise. All right, and then once you have this, I think when the examiners see this, they will say, "Oh, okay, this is nice. You know, I'll give you marks when this. Okay, 1 comparison, 2 comparison, 3 comparison, 4 comparison, 5 comparison. Yay! Then one more here, similarities, 1 mark. So total you get 6 marks in total. Done. Easy peasy, lemon squeezy. So try. I would say try to just do it in a short and simple way for the examiners to understand, right? So that you don't have to squint your eyes just to look for your answers in a paragraph form. All right, that would be a very good thing to do. So we've done this, no problem. Let's look at the next one. The diagram below shows a karyotype obtained from amniosynthesis and a karyotype procedure. So we have all of this, and you realize, like I showed us now, supposedly you're supposed to have only 2 copies of each chromosome. So this is a girl actually. Why do I know this is a girl? Because you can see here it has 2 copies of X chromosome and none Y chromosome. So this is here a girl. But this girl has Down syndrome because it has an extra copy of chromosome number 21. So this is the highlight over here. All right, study the information in the diagram below, above, and explain the karyotype procedure briefly. Your answer should be based on the following aspects: the purpose of the karyotype procedure, steps in the karyotype procedure, name of the disease the fetus suffers, and its gender. Very easy. Let's answer number 1 and number 3 first. All right, like I said just now, what is the purpose of the karyotype procedure? To determine, I would say that to determine the genetical abnormality. Or you can just say to determine the gender and the presence of chromosomal mutation in a fetus. That's the answer. Okay, 1 mark done. And then the third one, what is the disease and the gender? So what's the disease and the gender? So like I said just now, how do we know? Because has 2 X chromosomes. And like I said, it also has 3 copies of chromosome number 21. So therefore, it has Down syndrome. The fetus is a girl. Yeah, put female also if you want. Okay? It's a female with Down's syndrome. Okay? It's a female with Down syndrome. The reason is because why? We saw got 2 X chromosomes. And Down syndrome because it has extra chromosome number 21. All right, now steps in karyotype procedure wise, I'll give it to you later because that's quite an elaborated answer. So I'm just gonna remember this later. I'll pass you the procedures. It's basically very easy to understand. The amniotic fluid of the fetus has to be extracted out from the amniotic sac. All right. So what I'll do is that they will basically place a very, very long needle, all right, into the amniotic sac, and they will basically pull the syringe from the back, and they will try to extract some of the fluid inside. So the fluid inside, right, the amniotic fluid, may contain some fragments of cells from the fetus. And when they can take out the cells from that amniotic fluid, they'll be able to then put it under the microscope, and they'll be able to determine what is the chromosomes that this fetus has. Then after that, they'll be able to basically map out the karyotype, and then they will basically observe it and see if there's any kind of abnormality inside the fetus. Okay, so then after that, you'll be able to see it, and you will know if there's any chromosomal mutation, or even to determine the gender lah. If you have 2 X chromosomes, it's a girl. If you have XY, means it's a guy. That's it. Okay, nothing too difficult. But I'll give you the steps later on. Right. So I think that's all. That's all for this practice. So next week there is no class. Yeah, by the way, just say there's no next week no class because we're having a short Raya break from the 11th to the 17th of May. We will continue class right after that week itself, the following week lah. And then after that, we will start the new chapter. Okay. All right. So before we go, is there any question you want to ask? Yeah, all good, huh? All right, cool. So for those of you who celebrate Raya, I wish you a Selamat Hari Raya. For those of you who do not celebrate, happy holidays. And yeah, get plenty of rest, stay safe, right? Because there's a lot of cases of coronavirus right now. Hope you guys are at home. And yeah, do your revision, and I'll see you guys in the following week. Okay?